Welcome to Unstoppable Stories That Move. I'm your host, Sally Head Dahlquist. I've traveled all over the world to run marathons and am fascinated by the ordinary people who do extraordinary things. They just won't quit. No matter what life throws at them, they keep going. Mile after mile, challenge after challenge, they find a way to push beyond their limits. So listen as these seemingly normal people share their stories of resilience from the race course and beyond, and inspire us to keep moving forward too. Let's get to it.
Hi, Mike. How are you doing?
I'm doing well, thank you.
Good. Hi, everyone. I'm Sally Head Dahlquist with the podcast Unstoppable Stories That Move. And my podcast is actually the result of a charity that I started to raise $1 million for medical research. And part of the reason we want to raise money is because we know there are some really cool projects done by people like Dr. Mike Olin here that deserve more funding. So along with some of the NIH budgets that have been cut, we're here to draw attention to his laboratory, raise some money, and understand what he's doing, why he does what he does, why he is so unstoppable about the work he's doing in cancer research, and what we can do about it. So if you think to yourself, it's not like I don't do anything important. It's not like I'm trying to cure cancer or anything. But if you donate money to help cure cancer for people who are doing the work like Mike, then you can say, yes, I'm helping to cure cancer. I'm part of the cure. Donate, donate, donate. So let's hear more about Dr. Mike. He's unstoppable.
Unstoppable. Well, thank you for taking your time to talk to me. It's wonderful to tell my story. It's kind of a long, drawn-out story, but basically, after high school, I ran away in the Air Force.
You were escaping to
I was
Minnesota.
escaping to Duluth, Minnesota. The weather, it's not nice. So during my time there, I was married. I got married in Madrid. We bombed Gaddafi and killed his daughter, unfortunately. And they sent me to Turkey. And we went to Turkey, and we're out in the town, which technically we weren't supposed to be, but they served beer at the stores when you go shopping. So we did a lot of shopping. And
it's always about booze and
yeah,
girls.
of course. And for some reason, we started talking about the children there were the lack of, and it turned out there was a TB meningitis endemic or outbreak in that area. And over time, I just felt really bad. So I left the military for basically that reason, to come back and go to school. Came back, did a stint in Duluth, Minnesota again.
UMD.
A little plug for UMD. It's the best place in the world to go to school because there's nothing else to do in the winter. Otherwise, I would have been sitting on the beach.
In Madrid.
Yeah, well, yeah, or in a nice place.
Yep.
So did a stint, got my biochemistry, chemistry, and my hydrogeology degree was my kind of stress relief, little playing. Of course, my son was born during that too. So finished that and came to Minneapolis, joined industry for a while, thinking with these degrees I can do what I want, and quickly found out that's not true.
Right.
Got started working at a master's of virology in St. Paul campus at the Twin Cities campus under Dr. Molitor, Thomas Molitor, and he thought I was doing an outstanding job and got a chain grants and got my PhD in infectious disease.
Yep. And you and I met many years before that through our mutual friend, Suwan Tuwan.
Yep.
You had a company called Cell Technology, and now he's back here at the University of Minnesota doing research, working with you and your new group. And it's amazing all the different work that you're doing. So when I had my own company, Immunochemistry Technologies, you were working on the side. You had a little side gig doing quality control for us in the laboratory, helping us develop the products and getting them out there. And ultimately, we have, I think at this point, like 3,000 scientific papers using the products that my company developed. Oh, and something super cool is in Mike's lab here, thick copy of my book.
That I wrote. A chapter.
Apoptosis and cancer.
Yep.
That Mike helped write along with my business partners. Yep. I just did editing and proofreading, so I don't really get any credit. I was part of the chain to make this book happen.
Yes.
Apoptosis and Cancer, Methods and Protocol, edited by Gilmore and Aisha Alvaro. Get it today.
Yep. Page 101.
Page 109.
109. Sorry.
So if you need a page turner to help you sleep at night.
Yeah. Well, I've got plenty of those papers too. But during my, so working for Sally here was great. It helped me get through the low income of being a graduate student.
Yeah.
But I did a postdoc in TB meningitis working with my mentor who got me in this, Dr. John Olfest, doing some studies using his technology with brain tumors only on TB meningitis. And my mom, I got a call in June, and my mom had a stroke in Duluth, and I ran up there, and I'm like, that's a weird place for a stroke, and it turned out to be a breast or a kidney cancer metastasis.
So the kidney cancer is spread to the
Spread to her brain.
Yeah. So it's still technically a kidney cancer, but then it just moves around and starts living other places where it shouldn't be. And
so after 2 weeks after the funeral, I was going to approach John, but he came John Olfest, and he came up to me. He goes, "You ready to go to the dark side of neuroimmunology?" Because I was trained as a neuroimmunologist, and I'm like, I was going to talk to you about that. So I did a brief postdoc there, got a position here at the University of Minnesota, and unfortunately, the age of 37, he passed away.
Yeah, Dr. John Olfest
It was sad.
had cancer himself, one of the leading researchers, upcoming people at the University of Minnesota. A lot of people were.
It was sad.
And in fact, I noticed some of your equipment here says Olfest Laboratory on it. Continuing his legacy.
Yeah. So we continued his legacy. And at that point, we finished a vaccine that we developed. That's actually why I got hired to develop a vaccine. And we started what's called a DIPG, a diffuse intrinsic pontine glioma in children.
So it's a glioblastoma. It's
a high-grade glioma in the pons of the children.
So the pons controls your automatic neural system.
Automatic neural system. Yeah, the automatic neural system. You're so cute.
You're awesome. I got a degree in genetics and cell biology. It's your autonomic neural system.
That's
so they control your breathing, your heart rate.
It's actually a death sentence for children.
Yeah.
It's a horrible thing. And we noticed this suppressive protein that's called CD200, or also known as OX2. Was at high concentrations when our patients recurred in their serum. And that kind of like threw me back. I'm like, well, what's going on there? And no one really knew anything other than it was extremely immunosuppressive. It could shut down this protein.
Yeah.
And it's shed by cancers.
Yeah. So when people get cancer, the cancer itself can cause your immune system to shut down. It hides from your immune system. And that's how some cancers grow. For example, when people smoke cigarettes, the toxins in the cigarettes actually suppress your immune system. So every time you smoke, every time you take an inhale, you're giving yourself cancer, but you're relying on the strength of your own immune system to detect every single one of those cancer cells.
And that happens daily. And that happens a lot.
Yeah. Yeah. Your cells are always mutating, turnover, and growing. You don't need to help them mutate. But
we do.
But we do, fun things. I'm guilty too.
Yeah.
Yep. So finding tumors try and find a way around your own natural immune system.
And they're very good at it. They secrete a lot of molecules like cytokines and proteins that literally put on the immunological brakes. And this was actually around the time when the big breakthrough on immune checkpoints, that's what starts and stops the immune system, puts on immunological brakes, was discovered, especially by Dr. Allison, when they developed these antibodies against CTLA-4 and PD-L1.
So you're talking about CTLA-4
Immunotherapy,
and PD-L1. What molecules are those?
So these are molecules that will stop the immune system. They'll shut the immune system off. And like PD-1, PD-L1 is a ligand that's secreted by tumors, and it binds to its receptor its way to bind to the cell and shuts off the immune system. So this actually was a huge discovery. I mean, immunotherapy for a lot of solid tumors was stagnant until this time, and then it really took off.
And so what you're doing is also creating a vaccine against the tumors.
So what we did is others discovered the CD200 immune checkpoint, but it had two functions. It had one receptor that bound to the protein that stopped the immune system. It shut it down, completely shut it down. And another receptor, but no one knew what this receptor did.
So when you talk about proteins and ligands and antigens, what he's really talking about is kind of a lock and key system of your cell. So all of these proteins that go around, like if you're allergic to something, if you're allergic to pollen, think of the pollen as the key. And then your cell that it gets into, it irritates, has the lock. So the pollen key goes into the lock of your cells and opens it up and gives you allergies.
Does this job.
And it's the same thing with all the rest of the proteins in your body. There is a matching lock to match the protein. That's how proteins get into cells. And so you got to kind of go up there and knock on the door, put the key in, so the cell opens up and it gets inside. That's how some immune molecules, that's how growth factors work.
That's right.
And that's how your cells grow and live and take out the garbage and pee.
So this receptor, they called it an activation receptor. And now this was never described. What bound to that, what the key is to that wasn't known. What it does if there's something that binds to that wasn't known.
Okay.
So we're like, well, let's look at this. Let's see what's going on. Now, the problem with these immunological checkpoints is it's like, think of it like an airplane. Okay. So you're flying at 40,000 feet.
40,000.
Or whatever.
That's pretty high.
Yeah, that's a good airplane. And the hydraulics go out or the electricity goes out. It has 7 or 8 other redundant ways of rerouting electricity because you want them engines to stay high. They don't float very well. So that's the same with the immune system. CTLA4, PD1, PD-L1 system, CD200, there's other ones as well that are capable of stopping the immune system. So if one doesn't work, the other one does.
And that's what the tumors
right.
release.
And the tumors release multiple factors.
To try and stop your immune system so they don't grow.
So when Dr. Allison discovered the monoclonal antibody, it only does one of them. It only blocks. That's all it does is it blocks. So they had started coming in with multiple ones, looking at CTLA4, PD1, or PD-L1, blocking them. And that's when they really started to work, when they blocked multiple ones. So it's a fantastic system. Unfortunately, it only works on selected tumors, and there's a lot of toxicity or adverse effect. But it's great. I mean, they made such a big influence on immunotherapy. It really took off. Well, we started looking into the CD200 activation receptor instead of just blocking the inhibitory receptor. And it turns out that after years of struggling and trying to figure out what this does, it turns out that peptide, or the drug that we developed, will bind to a complex of these receptors, because there's more than one. And it'll activate and make the immune system in a configuration where it's ready to go ahead and attack tumors.
Okay, so the protein that you developed works on multiple receptors.
It works on one receptor. Well, multiple activations.
Multiple activation receptors that turn the immune system back on. They get it ready so that if something else comes in, the immune system is going to start working again to attack that tumor from within.
Right. So it prepares the cell. It turns it into a configuration that it's ready to go attack. But some people call this an adjuvant because it activates the cells, but it's a little bit more specific than that.
So an adjuvant is when you get a when you get a vaccine, the liquid that the vaccine is in is an adjuvant, because when you stick the vaccine in, all the other water,
It wants to turn on the immune system.
helps the immune system turn on and activate that vaccine, so your body reacts more to that vaccine.
Exactly.
So you want to stimulate it to work better.
However, unlike any other drug that we know of worldwide, this is the only drug that really does this.
That really stimulates the immune system.
A lot of drugs, adjuvants was stimulated, but this is more of a specific stimulation. However, we discovered that it activates a pathway so we can get rid of, downregulate, or remove all the different immune checkpoint inhibitory molecules like CPI4, PD-1. So now, not only is the immune system revved up for a specific anti-tumor response, it's protected because the tumors can't secrete or produce these inhibitory molecules, PD-L1, CD200 protein, etc., to shut the immune system down. So it shuts down all of them, at least the main ones that we've studied thus far. There's more that we're looking at. It shuts them down simultaneously with activating it, so the immune system's protected from the immunosuppressive tumor microenvironment.
Oh, that sounds very technical. So basically, there's a chain reaction in those cancer cells.
No, in the immune system.
In the immune system, so that it stays active. The cancer cells
it doesn't allow the tumor to put on those immunological brakes anymore.
Yeah, the tumor becomes powerless.
Powerless. So we like to say.
Powerless. Otherwise, the tumor is unstoppable.
Yep.
But you have stopped the tumor.
Yes. And this is the only drug that we know of in the world that targets this specific activation receptor and that does this.
Yeah. And
this is pretty exciting.
That's amazing. And it's a breakthrough in cancer research overall, because once you can use this technology for some of the glioblastomas and other tumors, it can be applied to other cancers as well.
That's
because
right.
It's such a basic pathway that you can figure out this chain reaction by elsewhere. And now you've found that it also seems to work in metastasized breast cancer.
Yes. So what we discovered, and this is more recently, when you're looking at CNS cancers, they're very difficult.
CNS is central nervous system, brain system.
Brain cancers, they're very difficult. In contrast, or unlike the brain, the periphery, basically everything from the neck down, has a different immune response than what happens in the brain. So even different locations in the brain have different immune responses. And what we discovered is that using different formations of this drug, what we think, we're working on this now, will find the key, will bind to different complexes of these activation receptors. And it'll elicit a different immune response. And we've shown that.
For peripheral.
So we have one that works in the breast. We're really focused on breast right now, triple negative. I'm on part of the intracranial metastasis consortium ran by Duke and WashU and Michigan. It's global. It's about 140 people strong now.
Yeah.
And we're looking at triple negative breast cancer. It's a form of breast cancer that goes into the brain and there's no cure for it.
Yeah.
And we discovered that one formulation of our drug works good in the breast. If we get in our murine studies, about 80% efficacy, or works in 80% of the time.
So that's the breast cancer cell.
In the breast.
But it goes to brain.
But this specific drug doesn't work in brain tumors. Otherwise, we have one drug, which is in the clinic now, I'll talk about a little later, that works on brain tumors, but it doesn't work in breast tumors. Okay. And that's why they elicit different tumors. So now we're trying to understand why. And it turns out that if we use the one that works in the brain, once that cell, the triple negative breast, goes into the brain, we actually can eliminate 100% of the time in our murine models, in our mouse models.
Okay, so that's the main point. Like, kind of lost in me. So you're developing this
It's a different form of the drug
a different form of your drug
that can elicit a different immune response if it's in the periphery
if it's in the body.
versus if it's not in the central nervous system
Right.
versus the central nervous system.
Okay, and one of those breast cancer tumors,
And
with that triple negative breast cancer tumor,
which go into the central nervous system.
yeah, it metastasizes. It starts to spread. It's a terrible disease. And as soon as though, when you find it in the brain, you notice that your peptide, your drug that you're developing, stops it from growing.
Correct.
And it doesn't stop it from growing. Does it shrink?
It shrinks. It disappears.
It actually disappears.
We've had
That's amazing.
in Washington University, St. Louis, WashU.
Yeah.
They did organoids, breast organoids, and they got, I think, like 75% of the tumors when they're in the cerebellum, in the lower part of the brain, disappear.
Wow.
And another cell line here, where we put it where the gliomas are, we have 100% disappear.
That's a game changer.
Yes.
This is the man who is literally curing cancer. Support him.
Working.
He needs a couple bucks. So donate. Donate now.
But these are on log halls.
Right.
We discovered the CD200 protein, the inhibitory protein, back in 2012
Right.
or 13.
And now it's
And that's where it
2025.
started. And it's really, when you go back and look at it, it was really funny because we would figure something out and then we figure out the next thing, and we figure out the next thing, and then we would stop and raise $30,000 a year, $50,000 a year.
Yeah.
I call it cookie and car wash it. That's what we did, cookie and car wash it.
Yeah.
And then we'd put in for small grants, and then we'd go on. And what we did was a little bit abnormal. In order to get to like a lot of NIH level grants, you need a lot of mechanisms. And I wanted to treat, especially kids. I just wanted to treat, we'd sit there at night just dreaming of this moment, actually.
Yeah.
And so instead of doing all the very mechanistic studies, which we're doing now,
To understand why it works.
we got with Chris Moertel, who we'll be talking to in a little bit, and we did all the things that we needed to translate this.
Into a useful clinical drug. So that patients can actually use it.
We did. We started treating adults with recurrent glioblastoma, and we saw that actually half of them had a very nice clinical response. With one of them lost the tumors, but it stopped the tumors within a month. It stopped them from growing.
Wow.
And we noticed in those patients that they had very nice immune activation. And all their inhibitory checkpoint molecules, PD-1, PD-L1, CTLA-4, PD, and
I got to write these down again. I'm
and
losing track of all
the
these molecules.
inhibitory CD200 receptor were gone. They weren't on the cells anymore.
They disappeared.
And the suppressive cells, like myeloid urine suppressor cells, T regulatory cells, they were gone too. They downregulated them. And this population actually lives significantly longer than the other population.
So these patients with glioblastoma, just death sentence.
And luckily, and the FDA allowed us to start a pediatric trial treating young adults with high-grade gliomas and what's called diffuse midline glioma, which is in the midline, but also includes DIPG, which we talked about. And that's ongoing at Children's Hospital right now with very encouraging response.
Children's Minnesota and Minneapolis. You have a partnership with them. And so you're actually treating children right now. Right. You're starting a clinical trial. You've had, 6 kids, and 4 of them are still on the study. And they're living longer than they...
Well, it's an ongoing trial with very encouraging response.
So this is considered a phase 1 trial where You just kind of want to get some preliminary data to see if it's really worth pursuing.
So
And right
what
now the
we're
answer is
doing
yes.
is called dose escalation. So we're trying to find the maximum tolerable dose.
Yeah. So what?
And
patient
right
handle?
now we're on our third dose, the highest dose. We just recruited one. Hopefully it'll be starting soon. But so far, we've seen no toxicity.
So the patients
It's
are
well
getting
tolerated.
additional
The kids are doing really well.
because everybody knows that chemotherapy itself is so hard on your body
Yeah.
because chemotherapy normally targets fast-growing cells, like your hair. That's why it falls out. And cancer cells also like to grow very fast. And so if you could kill all the fast-growing cells, kill the tumor cell as well as a whole bunch of other cells in the rest of your
You
body.
make it also.
Oh, no. Yeah, you know. And if you can find a drug to target the cancer that's more and more specific to that cancer, then you don't have that additional side effects of what's going on. You want it to be as targeted as possible. You want to find those molecules like CD200 or your peptide.
And there's other
Peptide
stuff.
and a
Well,
receptor that work together. So instead of having the bad stuff, you want the drug to go into that receptor, go into the cell and kill it. It's pretty amazing. I can really see your passion in this.
yeah, no, I am. I mean, it's been 12, 13 years of
13 years.
sitting on the
And
bench
you
dreaming of getting this in. And, you know, I honestly have to really thank foundations like Sally's Foundation, the Shavers Cancer Research, the Children's Cancer Research Fund, as well as Humor to Fight the Tumor, other funds like that, because we wouldn't be here.
This is where your money goes.
This is where the
When
money
you make
goes.
a donation.
Because to
It's
get an
laboratory.
NIH grant is they don't fund many of them, and it's very difficult.
Right. And right now we're in a situation where
They're
we
closed.
we need a balanced budget. So the NIH has made some hard decisions to cancel some budgets or cut the budgets, which
Oh.
I'm sympathetic for, having basically been bankrupt most of my adult life. You have to make some hard choices.
And they're only
have to-
funding. Over the last year, they've been funding about 9% of the grant.
9%. Yeah.
So all these thousands of people worldwide put in these grants, they fund like maybe 9% of them, 9 to 12.
yeah. And
So it's very, very difficult. So over the last 12 years, we get to a certain point and we start more cookies and car washes, raising money through little shavers. They put in every year, I think, with the exception of one, they've donated to this project. And then other foundations, that ABTA actually got me started. I got a discovery grants, and that's using that $50,000 grant. That's all I had at one point, is when I discovered the CD200 program.
And right now, during the government shutdown, well, in the budget cuts, you found out that June 1st, you're out of money. June 1st of 2026. And so it's a pretty urgent need right now.
Yeah.
And the reason I'm so passionate about your project, too, obviously I've known you for 20 years, even though we don't look any older than when we met. I know how good a project
do.
you're serving. And it's one of the projects that I would like to see continually funded. Like if if something's important, you should reach out and do it. Instead of complaining about the government shutdown or budgets, we're actually doing something about it. Hey, trust us as the steward of your money. Here's a project that has implications for all cancers, trying to understand the basic biochemistry of why it works and why your immune system sometimes is tricked, why it's fooled into letting these bad cells grow when they should be naturally dying.
And another thing that we did to raise money, that because these phase one trials are very complicated, is a number of years ago, in 2016 actually, I got with Dr. Moertel, and then I hopped on a plane to California to talk to one of my best friends, Suman.
Suman.
And we opened up a company called OX2 Therapeutics.
Yes.
And OX2 is CD200.
Yeah.
Another name for CD200. So we hired a CEO or CFO, Jeff Leiter, and we raised a little over $3 million through philanthropy. And that's how we got the clinical trials.
Dr. Ann Bendel of Children's Minnesota is taking care of the patient.
Is the director and the PI of the clinical trial.
She's the principal investigator of the clinical trials. And what is Chris's role?
Chris is the company's chief medical officer.
Oh, okay. Yeah. So there's two sides.
And so the three of us were the founders.
Yeah. And when you work in a laboratory like the university and you developed a technology, you spin off.
Yeah.
You can form a separate company to commercialize that technology so it can get into the hands of people who need it. Because the university doesn't. Yes, the university wants to educate people.
Well, they also
not
want
business.
ownership. Too.
Yeah, they want ownership. You have to give them a percentage. That's not unusual. Nothing that you're doing is unusual.
No. They spin off many companies.
This is the dream, the golden career of a research scientist, and having such a profound impact on human health. That's the dream. And you're unstoppable. You're not giving up with all these obstacles.
Been a long haul.
Yeah.
Been a long haul, but worth it.
Oh, thanks for letting us be part of it.
Oh, thank you.
So here with
you want to grab a chair?
Mary Teagles. Well, we'll just see how this works. We'll see how this works. And she's helping us raise money. Yeah. And helping us film this podcast and everything. So
I'm on board because I'm just in love with people who have missions that align with everything. So one of the questions I have is that both of you have been told many times by funding, by medicine, by life. What made you decide to do it anyway? What makes you unstoppable?
Stubborn.
he's stubborn.
I'm stubborn. I'm committed. Stubborn. You're here 7 days a week. I had a family. Well, half a family.
And you just got remarried.
I just got finally, even old people can get married. I did. I went through a divorce, lost my wife, and she's still here in Minneapolis.
Yeah.
And I was a single parent for a number of years. My son's best friend, this is probably stupid on my part, but my son's best friend, father was incarcerated and mother was having major issues that I don't want to talk about. So I took him in for 2 weeks and raised him as my own.
Remember that, yes.
Raised him as my own. He finally moved out when he was 19 or so, and he's been my son ever since. I have 7 grandchildren.
Well, 7 grandchildren now.
Between the two of them. Yep. So I was a single parent of 2 14, 15, 16, 17-year-old kids. It was a challenge while I was a postdoc.
Yeah.
But you sit here day and night just dreaming of this. And I go home on the weekends. I deliver pizzas to feed the kids. And you just do it. You just do it.
Because every time you look at that microscope, you look at those cells, you can tell it's working.
Yeah. And once
figuring it out.
Once you see animals living, it's pretty exciting. And then, a PhD, I always like translation. I always wanted to help kids. I wasn't smart enough to become an MD. I didn't think. Probably not. But I wanted to work with wanted to work with patients. So this is the closest I get to do it. But it's fine. It's great. I still have interactions with the clinical trials. I know what's going on. Every now and then I'll meet one. It's fantastic. And you do things for 12, 14 years thinking about now, and you just say, I'm going to do it. So when I discovered this, the activation receptors, even my mentors said, well, why you just put antibody over the inhibitory one? I'm like, well, that's kind of boring. And
everybody does that.
Everyone does that. And so I showed them it really doesn't work well. And, people tried to steer me a little bit different direction. So I just showed them, this is the way to go. I just saw the stubbornness. I just kept going. And, when you work on these NIH grants for, months on end, like 5-6 months, 40-hour weeks, working on them, and then you get them back and not discussed. Or, I got responses like, we cured dogs. We did a huge dog study. We had dogs literally on a plane from London to come to Minnesota. And we had the best responses for high-grade gliomas, glioblastoma dogs. Yeah. These are natural tumors. Nothing could touch them until we made the peptide.
So it worked in dogs.
So it worked great in dogs. And then you see people be excited. The reviewer's like, no, dog's not even. Okay.
So dogs are a little different, but
then we got into people and are dog recurrent, and 50% responded, and this is normal. And what did the reviewer say? Well, why didn't the other 50% respond?
Right.
I'm like, are you serious? So it's depressing. You work so many hours nonstop all night. And, you're burning the candles on both ends. You just get denied. And it's depressing, but, out of stubbornness, and I just kept going.
Right. And that 50% number is both good and bad, because you want people to respond, but everybody's different. So some people, that 50% may respond really well, and the other 50%
that's
doesn't.
a high response.
Yes. So how can you identify those patients that are going to respond and give them the drug?
Yeah.
So like the more drugs that are out there, doctors will try everything, and they try the most obvious ones first, the ones that work on most of the patients. And then they go down the line till they find the ones that work on more rare people, or, because
well, this disease, glioblastoma, is basically unstoppable.
Yeah.
Once you have it, it's going to come back.
But you're unstoppable in curing it. And you're a great example of the research scientists that are very specific on a specific biochemical pathway or a molecule that you're studying, in your case, generating these vaccines and immune responses against cancer, that it's a very narrow field of study. So you have somebody who's an expert in this particular area, and then you have people who are experts in adjacent areas, and you all come together at these meetings, like you were talking about the cranial cancer meeting, and talk about it and figure it out. And this is how it works. And this is why it's great to have a mix of different people, different projects.
Oh, absolutely.
Working all the time.
And you can't do it with all different people. Like, Dr. Moertel knew what to do in the human side. You can't know everything. So I consulted people for the signaling, for the mechanism. And, it takes a village.
It does take a village.
I did talked in a cancer foundation group in Duluth, actually, a few years ago, which is great because my mentors were there from undergraduate. And the theme of it was, it takes a village.
Yeah.
And it really does. One person doesn't know it all. You have people who knows how to do these newer spatial transcriptomics. I'm a neuro-oncologist, so I do the immunology. And then we consult with the smart people like the MDs that know what you need to do to translate it, because the FDA doesn't care how it works.
Right.
They just want to see it safe.
Right.
And hopefully it works. So I'm a PhD. I want to know how things work.
Yeah.
So it was kind of a pull, like, okay, I want to do this, but I want to figure out why. But to get into the clinic quickly, we kind of waited on some of the mechanisms. And that's when we started discovering that we could treat breast metastases. And we figured out it's two different signaling pathways between the two different peptides or the drugs that allow us to treat peripheral melanoma. We had some real good dog responses with the Mayo Clinic in the melanoma study, and breast. Those are the two big, other than central nervous system ones, that we've been looking at. And so the more we learn about the mechanism and how it works, the more we can actually use it for other than brain tumors or other tumors in the central nervous system. Because I think, I don't know, you can ask Dr. Moertel, but other than Jimmy Carter, I think most people pass away really quick once.
Yeah, he lasted
a
long time.
Yeah, it was surprising.
Jimmy Carter, Alex Trebek lasted a long time
Yeah.
with pancreatic tumor.
Yeah, I don't know if that was in the central nervous system.
Yeah, right. But sometimes people do live.
But I'm talking about once it goes into the CNS, it's usually.
So we're in the lab here, and we were just Dr. Moertel just walked in.
Mike, you've mentioned that your research you're doing is a breakthrough. We're asking people to find unstoppable. What is it like to be part of a team that is leading these breakthroughs and the impact that it's going to have on future generations?
Honestly, don't know how to explain it. It's so satisfying. I mean, I keep telling my kids, if we save 10 kids' lives over time, I did something in life. My place here is fulfilled. Obviously we want that.
Right.
But so when we're talking about these breakthroughs, it's a feeling that I as a PhD, and I don't work with people, it's just very, very satisfying. And I could see how MDs, the people who actually work with the children and adults, how satisfying that could be when their patients tumors disappear or even extend life. I mean, if we can extend life for these children, a good quality of life is important. Who wants to live in there 5 months and be sick and not be able to walk? And it's just the quality of life is bad. It's not worth it. But if we can extend life and extend the quality of life, it's just outstanding.
Yeah, so when I was sick as a kid, as a patient here at the University of Minnesota, not fun. My time here was not fun, but it was very effective. I was basically here. And the thing that really would depress me was the fact that my life didn't matter. I was going to die before I was 15 and no one would ever know. And now through my job and people like Mike and helping do the funding, wasn't going to get all teary. We can matter. I can matter after all, even if I don't have my own kids, don't do anything. It's great. So thanks, Mike.
Yeah, I'm taking it.
Well, Dr. Moertel just walked in, Dr. Chris Moertel. So maybe you want to come up and join us.
Sure.
Have a little seat behind, see if we can squeeze you in. So thanks, Dr.
There
Moertel,
she is.
for joining us.
You bet.
And then I'm the pop-in. I'm the pop-in guest. So producer, Maddie. Do you have some questions for Dr. Moertel?
I do. You are just part of an incredible team. And you're part
I know that.
I know. And the work that you guys are doing is just amazing. I'm sure every one of our lives have been touched by it in some way, shape, or form. Would you explain your background and then your role on this team and how you were introduced and why?
Yeah. How far to go back in my background? Well, I was the middle school science teacher, and one day one of my students' parents said, "Hey, Chris, have you ever thought about going to medical school?" And I thought about it for about 2 seconds and said, "Yeah, let's do this."
But on his behalf, his father was the director of neuro-oncology at the Bendel.
Not neuro-oncology, oncology.
Just oncology.
So,
it wasn't that specialized.
Yeah.
But I watched him and saw his life and thought I could never do that, thinking that he was the typical physician, which he was not. He was way above. And so trying to walk in his footsteps is a waste of time. I try to walk in his footsteps and I fall in, and it's really hard to climb out. So I've kind of made my own career, and I decided very early, I wrote on my medical school application, I want to be a pediatric oncologist, because I saw the work that they were doing here at the University of Minnesota, the first bone marrow transplant, cutting edge chemotherapy, the beginning of cooperative groups. This place was St. Jude, back before St. Jude's, and it was an incredible thing to watch. So I was very fortunate to be educated by those people early on. I went on to train at Texas Children's in Houston so I could get a broad breadth of pediatric training, returned to the Mayo Clinic for my oncology training, and then came up here to the Twin Cities. I worked at the children's hospitals from 1990 to 2007. And then in 2007, I was invited over here to be head of neuro-oncology. And so I came here specifically to go above and beyond, to work on discovery for patients who really didn't have any good answers. And so my early associations were with people in the lab, people like Michael, John Olfest, David Largaspada, because I knew that they had the brains to move things forward and to translate things to the clinic. And so right away, very early after my arrival, we got a clinical trial going with the vaccine that Michael helped develop.
We talked about that.
Yeah.
Yeah.
Yeah. And that vaccine showed us signals, but we weren't quite happy with that. And I'm sure Michael told you about how he sat down with a case of beer.
Oh.
And
well, okay.
yeah.
Okay, now I got to tell the
That's a good story.
Now I got to tell the
But he didn't make it through the case of beer, thank God.
Yeah, so early on during this trial, I was like very scared because I was new in this area, and I developed the vaccine. It was great, but I didn't want to ask any stupid questions, And being infectious disease, it took me like 3 months, and I went to my boss, Dr. Olfas, and said I got asked a stupid question because I talked to Dr. Moertel and others, and they couldn't answer that. Why do we vaccinate where we do? And no one really gave me a good answer. And he looked at me, and this was a stocky guy. He was small, short, but big. And I was in the corner, and he's like looking at me like that. I thought he was going to leap over the bench and hit me or something, because why would you ask if this is stupid? And he's like, that's a great question. So I decided to do an experiment where I gave some mice tumors and other mice without tumors. And there was a guy from China, actually a neuro-oncologist. He was a surgeon with really good hands. And he got the serum. And I did this 3 times, did proteomics, looked at what's in them, put them in a spreadsheet. I was a single parent at the time, so I grabbed a case of beer and went home and looked from A to Z, interesting proteins or whatever's in there. And, well, thank God CD200 started with C. Yeah. Because I probably would, if it was Q, I would have missed it, I'm sure. But I sat there with a case of beer, and if it looked interesting, I'd look it up. And then I found CD200. I'm like, wow, this is really cool. And then we looked in the serum of our patients, and
yeah.
And more importantly, what kind of beer was that?
it was old Milwaukee. I was a postdoc. They were returnable bottles.
That's right. That's how we made it through college. We got Pfeiffer or Buckhorn and returned the case so that the next set of beers was
$8
cheaper.
a case. It was great.
Yeah, so good, so good.
So
Important part of everybody's education.
That's my beer stuff.
Yeah. But anyway, so Michael brought that to us, and that's when it really started, that we needed to find out what this CD200 was all about and how to deal with it. And Michael did a really eloquent set of experiments. We brought in collaborators from really all over the country to help to verify what he was finding. And we wrote our first article, and it really showed in that article where CD200 is important and how it's very important to understanding how brain tumors evade the immune system. And so Michael went on to invent the peptide that has now become a drug, and we have begun administering it to patients. And I'm grateful for Michael and his work, but I am grateful for our collaboration too, because I watched very carefully what he was doing and knew that it was something that we could bring to the clinic to benefit patients. And that's where we are now. And I know that he brought up earlier how we had to talk to the FDA and make sure they understood what we were trying to do. He had to fashion treatment protocols. And my specific interest is in helping children. But before you can help kids, you have to treat adults to prove it's not toxic in adults. So we did that. We did a small pilot trial here at the university treating adults. And then we brought the results of that trial to the FDA, and they approved the pediatric trial that we're doing today. And so that's kind of how it marched along. And, how lucky are we to be at the University of Minnesota, where immunology is really a core science. We lead the world in so many areas within tumor immunology. So the resources here are fantastic, and we were really able to do a lot. But, we realized the potential of this drug and realized that without some kind of commercial support, we really weren't going to get anywhere. And so we reached out to local business leaders very early to develop a company. And that way we could bring in capital to help to support.
Bring in money,
capital, money.
Donation. Investors.
And that helps to support Mike's research, and it helps to support the clinical trial we're doing right now.
Yeah, research is a global problem. How did I say this?
Funding research.
It's not a problem. Diseases.
Sometimes research is a global problem.
Cancer and these diseases, it's not just a local problem. It's not just in the U.S. It's not Germany. It's global. It's a global problem. Everybody has to help support it. And the University of Minnesota is one of the leading institutions to help discover cures and funding and understand what's going on. And so these diseases don't care what color you are, don't care where you live,
your age,
don't care how old you are or what your parents did for a living. You don't care how much money you have. It all comes down to basic human biology and biochemistry, and that's what we all share as part of humanity. It's a human problem.
Absolutely.
And another issue with what we do, which is a problem, is we're in a clinical trial, we're trying to get money, funding, but a lot of the big pharma, they don't care about these tumors because they're rare tumors. Not every, only 400 maybe children get DMG a year. So they're not a huge problem in large pharma's eyes.
Right. Yeah. It's often considered an orphan drug.
Making an orphan drug.
Such a small population. And what drug companies are doing is they talk about they're looking for targets. So they mean, what they mean by targets is some sort of biological molecule that affects the cancer cell that could be turned into a drug. And by discovering the CD200, you've discovered a potential target that can be converted into a drug, mass-produced, and used to treat people. And even though it is a small population, what you're finding is that it also affects other kinds of tumors, particularly when breast cancer metastasizes into the brain. You could look at that. So many tumors have the same basic biochemistry, and even though you're working on a smaller one, this ripple effect, chain reaction, really helps cure other cancers too. So tell us, where are you going next with your clinical trials? We've got 4 patients now, doing okay, and what's your next steps?
Yeah. Is it 4 or 6?
We had 6.
So we enrolled 6. We've got our next dose level to go. And then if everybody does well at that dose level, then we open it up and prove that we have our maximally tolerated dose, otherwise known as a recommended phase 2 dose. And with that recommended phase 2 dose, we can then go on to an efficacy trial. And that efficacy trial will look at large populations of kids with hard to cure tumors. And we're already talking to large collaborative groups about going forward with that phase 2 trial. It's in development. And I think you'll be talking to Ann Bendel later on.
Yes, we are.
And Ann is actively putting that trial together now.
Okay.
And we've been working with her to make sure that everything goes. So we're really excited about that.
So right now in your phase 1 trial, you've given the kids several doses of this medication, and now you're trying to give them a higher dose and see how they respond, and then add more kids. And once you determine that dose works okay, that it's kind of safe,
right?
Then you're going to start your phase 2 and add more patients. And
right.
50 or more patients, and that's going to cost how much? What are you looking for there?
Yeah. It costs millions of dollars, but the nice thing is if you've got a cooperative group working with you, you've got Dr. Bendel working with us. We already have a lot of stuff going on with our company providing drug. We're hoping that we can keep the cost of the trial down. And by doing so, we can get by, hopefully with less money. Many phase 2 trials cost $20 million plus.
Yeah.
And we're really hoping that we can do our trial for much less than that. But
Another benefit of sorting, especially with D&G, is the FDA doesn't require us, will require us to do a phase.
They won't require you to do a
So
phase
they-
3. So as soon as you're showing that these kids are responding and the drug is inhibiting, it's stopping this cancer from growing, you'll be able to start using it immediately after the phase 3. That is a process called what? Fast tracking. The
Yes.
FDA has some fast track where when drugs show particular effectiveness, that you can get them out there right away. It's amazing.
Yeah, and we've already received rare disease designation. They're ready to give us orphan drug approval once we get to the efficacy stage. And so we're in a really good place to get fast track. And so, that would benefit us, but as a company, but it would certainly benefit the people at the end who need the drug. So
Yes.
we're very hopeful that all that goes.
Who need the drug, you'll get it into those kids. And then meanwhile, continue your benchtop work with Dr. Mike here, Dr. Olin, to keep investigating how it could potentially work with other cancers and try and get it to work with the metastasized breast cancer as well.
Yeah. Yeah. And if we have success with our very rare disease, people will pay attention and use it for these more common conditions. And we've seen expression not just in breast cancer, but in melanoma, which also can metastasize to the brain, and in a number of other childhood brain tumors. So I think the possibility that this particular drug will benefit not just 400 kids, but thousands of people is very high.
Well, not only the drug itself, but the vaccine technology that you're using along with it.
That's what really excites me. Ask earlier, what excites me about this. This isn't a drug derived from a previous drug or another discovery that people know the mechanism. We are the only one in the world doing this. It's unique. Nobody else figured this out in the past. It's an actual discovery.
Yeah, pretty cool.
Do we have any more questions from our tweets?
Too. Over here, we were putting our heads together. 20 million sounds like a very high price tag.
This is Matt here.
20 million sounds like a very high price tag.
Sounds low to me.
Oh, where does that 20 million go? And when a donor says, does my $100 or $1,000 make a difference? How would you respond with that?
Well, number one, we're going to try to do it a lot more cheaply.
Right.
First, you have to manufacture the drug in a way that it's very safe. And so all that needs to happen so that you have a drug that you can actually put into people at low risk. So that's
Just
number one.
putting it in a vial is about
$120,000 to put it in a vial.
in a
Yeah.
vial. Just to put it in a vial.
Yeah.
That's not making it. That's putting it in a vial.
Yeah. Yeah. So, the steps to get there in a safe way are pretty important. Number two, you have to get patients, and sometimes you have to help them get to where they can receive these drugs. So people travel great distances, and we actually have, for this particular trial, we have a separate fund set up to support travel.
Yeah.
And lodging when they come from great distances. So that's important. And then the science is very important, supporting testing around how the immune system responds to this. Is there toxicity in the body? We have to do all that lab testing. We have to do radiology. We have to do MRIs on all the patients to measure what's happening with the tumors. And then we have to support all of the institutions that are participating in this research and their research infrastructure.
Yeah, and pay for the lab.
But what people don't often understand is, you could probably comment more on the price, just making sure that the stability of the drug is tens of thousands a year. We have
That's it. That's the shelf life.
The shelf
We want to make
life.
sure that it lasts.
it has to be tested every year. The we have to pay a biostatistician,
Biostatistician,
and we have to
big
we have to pay for a regulatory board. How much is that?
Well, all of that goes into the cost of research infrastructure, not just lab, but the people who are making sure that their institutions know that this is a safe thing they're doing for the patients.
Right. And those are extremely expensive. That raises that cost.
You're paying for people. Yeah.
Paying for people, you're paying
And
for
they don't do, and they
technology.
don't do it for cheap.
Yeah. And then just
beer.
to transport our vaccine that works with the CD200, just to transport the vaccine from here at the University of Minnesota St. Paul campus to Minneapolis Children's
Crossing
each
river.
time. Yeah. How much does it cost for one process?
$1,000
$1,000.
$1,000
per vaccine. For vaccine.
Well, can't you
Because
just
it
stop
has
a
to
lift
be
for
in.
that?
Yeah.
Maybe they should start a new subcategory.
Yeah, but it has to be stored in liquid nitrogen and
Yeah.
transported over. So there's a lot of very high-tech things going on here that all become part of the action.
Yeah. But there are ways of making this better as well.
Making it better.
We discovered, and actually, we discovered this drug, as I said, developed, I use the A word, adjuvant.
Adjuvant.
I know.
We've
I
tried
always scold them for saying that. Activates the immune system, but you got to tell the immune system what to do. And in CNS tumors, they're called what's immunologically cold tumors. They don't secrete those antigens that you explained earlier. So we developed a vaccine, which we started with, a tumor lysate vaccine, to give these keys to the immune system. Well, we've been working on better ones. That's what costs $1,000 to transport. And a lot of facilities don't have the means to store it because it's in a vapor, what's called a vapor phase of liquid nitrogen. What we discovered recently, and we've been working with other groups, that this technique called focused ultrasound will go in and beat up the tumor and release these antigens.
So you can use ultrasound. You can use sound waves
On
on that tumor in the brain
Yep.
to break it up.
Exactly.
So
And-
you break it
and
up. It will
In our glioma mouse models, we get about 50-60% survival with the tumor lysate that we're doing now. We've got 80 to 100% survival using focused ultrasound.
Wow. So the tumor lysates you're talking about is when you take the
tumor-
cancer cell itself and you bust it up and you turn it into a liquid and then you
Give it-
re-inject.
Yeah, that's the vaccine for those antigens.
Yep.
And there's a group on the East Coast, Virginia Tech, that we're working with, and they want to start a clinical trial with pediatrics, high-grade glioma, using our drug with focused ultrasound.
So developing this new additional laboratory technique.
Yep.
So you've got a new drug, you've got a new vaccine, you've got a new ultrasound
Well, the drug
technology.
thing, the drug's the same.
Well, your drug.
Yeah,
It's
yeah.
not
My drug,
Voldemort.
but we'll be using focused ultrasound instead of tumor lysate. So that'll bring down the cost quite a bit. The cost of making the tumor lysate and storage, and just transporting it across the rivers, $1,000.
Yeah.
Well, imagine it's over wherever,
To Virginia.
Yeah.
Or
Yeah. And
German-
focused ultrasound machines have a cost, but it's a one-time
Yeah,
cost
exactly.
instead of paying every time.
And focused ultrasound's growing in clinical trials. They're using it more and more.
if you're taking advantage of any and every technology, new piece of equipment, new light, color molecules, all of that.
It's pretty fun.
Pretty fun. It's satisfying.
My
And
technician's probably
dreams come
laughing
true.
like it's not fun.
Well, we should probably wrap this up here today. It's been really fun talking to Mike and Chris. So can you tell us why are you unstoppable? What is unstoppable means to you?
Well, I've learned throughout my life that persistence pays. And yeah, it's a great question because I think it's how I'm made. You've got to kind of hang on to things and see them through. It's true here. We started with this discovery way back in 2010. Here we are in 2025. But it's because we've persisted and know that we've got a technology that we trust. It's the same thing when I was in sports. It's what I try to instill in my kids now. If you start something, see it through.
And didn't you just retire from the university?
I did.
a couple days ago.
A couple days ago,
And
yeah.
now this is your side gig.
Yeah.
Right? Your side gig.
Well,
is-
becoming less and less my side gig and my primary
It's your
gig.
full time,
Yeah.
and now it's your retirement job.
Yeah,
I mean,
yeah.
that's a big job.
Yeah.
Curing cancer is your side gig. And
Yeah.
retirement. Then, Mike, what about you? I know you're unstoppable. What does that mean for you?
Just the same thing. See it through, persistent, stubborn,
Yep.
Oh. stubborn.
He is stubborn.
I know, but I'm very passive aggressive stubborn. I'm not in your face and that will never work.
You must be from Minnesota.
Yeah. I mean, like, I'm stubborn in the sense that I'll prove to you this week better, either way. And if the other way's better, I have no problem.
So if somebody says to you, you'll never be able to do that.
Oh, I've been
you're
told
gonna show
that.
them.
I've been told that. And, you know, just keep going. But I also listen to people. One thing, my strength is X, not Y and Z. And like clinical stuff, I just listen to them and I don't try to chime in the other way, other than, well, can you try to do this? And let people do their job. I let him do his job and let the chief financial officer do their job, which is good because when they start talking about funding, my eyes glass over. And I don't want anything to do with it. So, and I listen to my technicians. And that's what really drove it is, you know, I'm not better than my technicians.
He's not better than his technicians. Did everybody know?
They just make it look like I am. But the point is they're here every day looking at this, doing experiments. They see something different. My technician, he's not. She's
And we got the lab manager here.
the lab manager. But I listen to you. Come on.
Come right here.
But I'll listen to them if they have an idea. I'll let them run with it, and we'll switch as you know. If they say no, this isn't working. It's like this. Great. Let's look at it.
So this is the woman, the hands-on lady who's working with all the tumors. And please introduce yourself. Hi. Okay. Well, hi. I'm Dr. Singh. I'm working with my child for the past 3 years, and yes, he's correct. Thank you. Thanks for joining us. And we appreciate you showing us around the laboratory today. So we should wrap this up. Really appreciate you guys coming out, sharing your story, your adventure with us. Great.
Thank you.
Welcome.
Thanks for your interest.
Thanks for tuning in to this episode. I hope you enjoyed listening to it and learned a few things. Well, the people that we talked about today, they don't quit, and neither should you. Whether you're running marathons, rebuilding your life, or just searching for that next spark, you are more capable than you think. If this episode hits home, share it, subscribe, leave a review. Let's build this community together of relentless people who refuse to stop moving forward. And if you want to do something even bigger, support the mission. We're raising $1 million for medical research, and your support makes a difference. Check out our website at unstoppablestoriesthatmove.com. I'm Sally Head Dahlquist. Until next time, keep running, keep pushing, and never stop proving to yourself what's possible.